fruit, fam. I think there's some things we need to get educated.
You know that's not biodegradable, man.
Wait, guys, you know that's not biodegradable, right?
Yeah, it takes like 1,000 years to biodegrade.
Today, you have to learn about PHB, Polyhydroxybutyrate. It's a bioplastic which degrades in 10 years. Maurice Lemoine in mid-1920s was the first person who discovered monopolyester of poly-3-hydroxybutyrate. Later on in 1980s, the Imperial Chemical Industries Limited was the first to do intensive studies on PHB in biosynthesis and biodegradation.
The use of BHP grows popular because it offers many advantages over traditional petrochemical derived plastics. Besides its complete biodegradability, BHP is formed from renewable sources. It has better physical properties than polypropylene for food packaging applications and is completely inert. The poor low impact strength of BHP is solved by incorporation of hydroxyvalerate monomers into the polymer to produce BHPV. It completely degrades into carbon dioxide and water under aerobic conditions. Microbiosynthesis of BHP is the best method for industrial production because it ensures the proper stereochemistry for biodegradation.
The main reasons for brittleness of PHB include the secondary crystallization that occurs during storage at room temperature. Also, it has a low nucleation density, therefore it forms large spherulites which causes cracks and splits, and this has a negative influence on the mechanical properties. Thirdly, it has glass transition temperature near ambient temperature. PHB is a partially crystalline material, has good barrier properties such as PVC and PET, and can be used in packaging industries as a biodegradable plastic for solving environmental pollution problems. There are many references to attempts to mix PHB with other polymers with the aim of improving its mechanical properties, unfortunately with only limited success.
Nowadays, plastic and synthetic polymers are mainly produced from petrochemical industry, which resulting in environmental pollution. Biodegradable plastics seem to be alternative choice for industrialized countries such as USA, Japan, and European countries because of the properties of the materials are suitable for a wide range of fields. Furthermore, more than 30% of household waste are all type of packagings. That's a big problem. That's why we have to use biodegradable plastic.
I think Mo did the same, man. I think Mo did the same. We're so sorry, guys.
Thank you, guys.
Thanks guys, thanks for understanding.
Appreciate that, guys.
Feedstock processing for PHB. Poly-3-hydrobutyrate is a bioplastic which provides an alternative to everyday non-biodegradable item polyethylene. PHB occur naturally in microorganisms as the carbon energy storage and electron sink, similar to starch. It can be most commonly found in soil bacteria, for example, Pseudomonas and corianform bacteria. PHB production in plants is another type where bioplastics can be produced directly from sunlight and carbon dioxide. For example, transgenic alfalfa has PHB concentration of 0.025 to 0.18% dry weight, as compared to that of sugarcane 1.88% dry weight. Recently, a hybrid Populus tremula X alba was genetically modified to induce PHB production. All bacteria capable of producing PHB do so during the stationary phase of growth, where cells are limited to a particularly essential nutrient in the presence of excess carbon source. During the initial balanced growth period, PHB is not produced, but cell mass is. When phosphate is limited in the second step, PHB is accumulated. This is because the organism under these conditions can no longer reproduce, but produce PHB as energy storage. By necessity, this process uses a defined medium containing glucose or sucrose, because unrefined feedstocks like molasses contain sufficient nitrogen and phosphate to interfere with the timing of the nutrient limitation used to induce PHB synthesis. The most common bacteria used for industrial scale production of PHB are alkali genes Eutrophus A. latus and Lactobacillus acidophilus. PHB production can vary depending on the nutrient limitation imposed during the synthesis of the bacteria. There are three main nutrient notation used these days to compare the activity of individual organism in such surrounding. In industry, to make the process more economical, it is important that the highest yield of PHB is produced. Ammonia limited, potassium/oxygen limited, or glucose limited cultures are examined generally. Eutrophes and Pseudomonas aeruginosa have a highest yield and PHB content when ammonia limited culture is used, whereas Azotobacter baijinii is maximum under oxygen limitation. PHB production may vary depending on the substrate used as the carbon source. Glucose, fructose, or milk whey are used commonly as they are renewable. Ammonium sulfate, ammonium chloride are other used as nitrogen source. It was found that glucose is often used in industry as it is cheap, thus economical, and also glucose provides with the highest yield of PHB which is preferred. PHB produced on the popular plants have to go rigorous treatment to obtain the desired PHB. The major task is to remove chlorophylls from the leaves and extract PHB. For that, PHB plant is grinded and mixed with ethanol. As ethanol is evaporized, chloroforms can be extracted, but the industrial scale of plants for PHB production is relatively low as major companies invest to produce PHB using the substrate method. Therefore, optimum selection of substrate and nutrient limitation would result in highest yield of PHB.
Hi, this section is about the fermentation production of PHB. In order to fully understand, we must first understand the basis behind fermentation. Fermentation is a natural process which has been utilised for many years in the production of wine, beer and yogurt throughout history. It is simply an anaerobic reaction done within many bacterial and yeast cells in which a sugar or starch are converted into an alcohol or an acid. It is done in an anaerobic environment as an alternative to respiration. Thus, cells will tend to avoid using fermentation in an oxygen-rich environment because respiration produces more energy than the energy-producing step glycolysis in fermentation. The common glycolysis reaction should be shown on screen now. We can see 2 ATP units are created per mole of glucose compared to the 38 ATP per glucose normally produced by aerobic respiration. Fermentation can be used in the production of PHB, and this occurs in 2 steps. The first is pre-fermentation, in which cells are grown at 30 degrees C in an air and mineral-rich environment using glucose as the energy source. The cell will begin to multiply and reach a maximum cell density of 20 grams per litre in 24 hours. The cells are then put into a sealed fermenter, where the cells will continue to multiply, reaching a maximum cell density of 100 grams per litre. During this process, PHB will start to be produced as small granules with diameters of 0.3 to 1 micrometer as a result of the cell using fermentation for producing ATP. Production will begin to drop as the pH becomes lower due to the hydrogen ions or due to a lack of glucose, leading to cell death. By the end of this batch reaction, the cells will have a dry mass of 80% PHB. A case study has been produced to understand the optimal conditions needed in the production of PHB. They looked at testing the effects of both physical and environmental conditions needed to maximize production. This included temperature, time left in the medium culture used. The data revealed the maximum PHB output to be 0.412 grams per 50 mole, or 8.24 grams per litre, which was achieved using the bacterial cell Lactobacillus acidophilus when grown for 4 days in the fermenter. The test also revealed the optimal temperature to be 37 degrees C, a pH of 5.5, and the nutrients were also considered. From the graph, it can be seen that a mixture of olive oil and glycine will give the optimal conditions for PHB manufacture. This would not be used in a large-scale production of PHB, as olive oil is expensive. Instead, glycine would be used. The mixture must be tested on a regular basis to test the concentrations of both detrimental and beneficial influences within the reactor. The reactor must also be checked regularly for any air entering the reactor through cracks or semi-closed valves, as this will affect fermentation rate. In order to regulate pH, a weak base must be used in order to maximize production through minimizing the risk of pH decreasing. As a result, pH will also need to be monitored.
Hello, this section is about the downstream processing of PHB after the fermentation process. There are 2 isolation processes for PHB. Firstly, mechanical loads destroy the cell walls and then the PHB is dissolved in chloroform. The remains of cell are then separated by centrifugation and then filtration. The second method is the enzymatic method, where enzymes at 37 degrees C are used to destroy the cell wall. There are 3 methods for the processing of PHB polymer that is extracted. Firstly, there is the plastic extruder. This process produces the polymer in pellet form, which is further used to produce films, bottles, and injection molded parts. The task of the extruder is to take the solid plastic and using heat, pressure, and shear to transform it into a uniform melt which can then be delivered to the next processing stage. The final melt must be uniform in both concentration and temperature. The extruder contains 3 sections. The first section is maintained at a relatively low temperature. The second section is where the melting occurs and pressure is built up. Finally, the extruder outlet is the metering section. At the end of the barrel, the melted plastic leaves the extruder through a die which has been designed to give the desired shape to the stream of the melted plastic. The polymer viscosity is dependent on both temperature and flow rate. Secondly is production of a cast film using a chilled roll. The extruder is shaped like a slit die, the width of which is much greater than the thickness. The cast film and sheet are produced by extrusion of the melt to chilled chrome rolls. The plastic exits in a downward direction towards the chill roll. Contact is tangent and the film travels in an S pattern around at least 2 chill rolls. The first chill roll normally operates at a temperature of 40 degrees C. An air knife is used between the plastic and chill roll. This leads to the formation of a film with better transparency and thus increases stiffness and may also increase output. The film's dimensions are controlled by the extrusion rate, melt temperature, cooling, and takeoff speed. Thirdly, there is the common process of injection molding. Injection molding is used for producing base cups for bottles, margarine tubs, containers, and packaging articles for the food industry. During injection, the plastic is melted in an injection molding machine. It is equipped with a reciprocating screw that moves backwards and forwards in the barrel. When a sufficient volume of melt is accumulated, the screw stops rotating and is driven forward mechanically or hydraulically, thus injecting the plastic through a nozzle into the mold through the spree, a system of runners, and finally into the cavity. Once the mold component is cool enough, it solidifies and the mold opens automatically. The cycle time depends on the size of the molded part and molding conditions. To be more important in the future to decrease plastic pollution, the costs of biodegradable polymers should equal that of conventional polymers, as well as meet their quality and processing conditions. At the present time, polyethylene and polypropylene costs approximately $1 per kilogram, but PHB costs $2 to $8 per kilogram.
Bioprocessing. For PHB synthesis, I'm going to talk through the synthesis of polyhydroxybutyrate steps and the development of microorganisms in industrial scale. Alkylagene eutrophus is a bacteria that is used in the industry of producing polyhydroxybutyrate. Scientists select gram-negative strains in order to optimize production yield. The synthesis of polyhydroxybutyrate involves glucose as a carbon source and 3 enzymes. First, 3-ketobylase, acetyl-acetyl-CoA reductase, and PHA synthase. The first enzyme, 3-ketobylase, is responsible for carrying the condensation reaction of acetyl-CoA into acetyl-acetyl-CoA. And acetyl-acetyl-CoA reductase reduces acetyl-acetyl-CoA into R3-hydroxybutynyl-CoA with an ADPH serving as the reducing agent. After that, R3-hydroxybutynyl-CoA is then polymerized into polyhydroxybutyrate with the aid of PHA synthase. According to high production cost of PHA bacteria, engineering study of pathway in eukaryotic cell system became very important. In order to reduce the production cost, a gene transfer of alkylagene eutrophus to the plant's Alacabadopsis thaliana can be done through genetic engineering. The technique is called gene splicing. Yeasts are more flexible than bacteria in terms of physiology because many of them have been approved by Food and Drugs Administration. Metabolic engineering is a practice of optimizing genetics process within a cell. This technique analyzes constraints of production desired compounds. The constraints could be released by using genetics engineering techniques, In order to relieve constraints, we insert new gene to improve production. For example, the transgenic strains Saccharomyces cerevisiae were able to produce PHA with higher yield on non-conventional transgenic strains than recombinant strains.
Batch and fed batch fermentations are widely used in the industrial fermentation process. Fed batch cultivation is more efficient than batch cultivation for producing high product and cell concentration. This is due to medium composition being controlled by substrate inhibition. Consequently, high initial concentration of substrates fed can be eluded. The downside of this system is the long downtime between two batches, resulting in high operation cost. The major cost in the production is the cost of the substrate. Thus, the selection of suitable carbon substrate is of crucial importance in determining the overall performance of the bacterial fermentation and the cost of final product. The choice of renewable, inexpensive and most readily available carbon substrate that could support microbial growth is suitable. Some options for carbon sources are waste effluents to plant oils, fatty acids, alkanes and as well as simple carbohydrates. Each year, a large amount of waste materials are discharged from agricultural and food processing industries, and these wastes represent a potential renewable feedstock option. Glycerol is seen as a good option because it is generated from a waste stream in the biodiesel process, effectively killing two birds with one stone. BHP's thermoplastic properties in a range of 170 to 180 degrees Celsius, which means that it can be processed with the standard production processes which are used in the plastic industry. However, it must be taken into account that molten PHB sticks to the skin. A further disadvantage is a greater tendency to crack and break compared to polypropylene, which is normal plastic bags. In order to improve its properties, PHP is therefore mixed with other components such as cellulose acetate, cork and inorganic materials to form PHP blends. The benefits of blends and copolymer are producing a product with a range of properties such as different melting points and crystalline structure. The use of BHP for plastics does have some environmental issues. Current technologies have impacts to the environment in terms of energy requirements, eutrophication and acidification. Once BHP is in an ecosystem, the increase of nitrate and phosphate promotes growth of algae. This then blocks sunlight from organisms in the ecosystem. Bacteria use up the oxygen, leaving the ecosystem lifeless. The other issue of the use of BHP is that it causes acidification. Acidic soils cause significant losses in production of agriculture, and where the choice of crops is restricted to acid-tolerant species, profitable market prospects may be reduced. Research is being done to improve this by improvements in the fermentation technology or changes in the feedstock.